So first, I'm actually going to turn it over to Shelley Welch. Shelley is the president of the WV chapter for ACRP.
Thanks, Debbie. Thanks so much for coordinating this presentation. I appreciate it very much. As Debbie mentioned, I am the president of the West Virginia chapter of the Association of Clinical Research Professionals. Connie Sorello is the vice president. She is also in attendance today. So if anybody is interested in joining the West Virginia chapter, really a goal is to promote research throughout the West Virginia state, and we do that by holding these events and training and networking as well. Unfortunately, because of COVID, no networking could happen today, but we wanted to continue to provide training. So thank you so much, Debbie.
Yeah, absolutely. Thank you, Shelley. So we are here today, we're going to talk about building a research protocol, and it's really starting with the end in mind. This is actually going to be presented by myself and Padma. Padma has been with WVU for 20 years, and yes, I did make her do that math. She is a certified clinical research professional for basic research, experience, and current expertise in clinical trials and regulatory affairs and human research population. So she is on the line with us as well today. So if questions pop up, I have several people from our staff that's developed protocols. So if you have questions, please feel free. You're more than welcome, please, as I'm going through slides, to unmute yourself and ask the question, or you can place the question in the chat box. I do have a co-host today, so if a question does pop up in the chat, Lisa will stop me just because chat is so busy. I just want to make sure we don't miss anything. And I'm actually going to go ahead and turn off my video just so it's not distracting as I present. So, all right, so let's go ahead and get started. So our objectives for today, first thing we want to do is understand when and why an investigator-initiated protocol for clinical trials is required. So we're really going to focus around that because obviously if you're doing an industry or sponsor or study, the protocol would be provided to you. So today's presentation is really around an investigator-initiated protocol. We're going to review how our protocol is helpful to researchers, discuss content expectations for sections of the protocol, and then also recognize protocol problems, spots, right, and ways to improve protocol writing. And we also want to identify resources to assist with protocol development, and also really understand when a data safety monitoring plan is required. So let's start with the end in mind. So the first question that you might want to ask yourself, is it reasonable? So what do I mean by that? Is it reasonable for a patient, to do a fasting blood, fill out a three questionnaire survey and go for an hour testing? Well, remember that patient fasted. Is that reasonable? Can they wait that long before they eat? So kind of thinking about things like that as you're working through your protocol, excuse me. Do you have the resources, right? Is it just you? I see there's a question. I don't know. They're asking if the slides can be any bigger. And I apologize, I don't know. Let me try one thing here and see if this does anything. I'm not sure. All right, did that help at all? Oh, okay, I see what's happening. So what you'll want to do is, I don't know if you have two screens. You'll want to, because you're currently on speaker view, so you want to turn it where you can actually see the slides. So you can actually turn off your speaker view. I don't know if that helped the person or not. So if you, in the right-hand corner above where it says speaker view, you should be able to either exit or enter that screen. And then that should be able, oh, okay, you got it. Okay, sorry, thank you. All right. And I'm sorry, Lisa, I took over your co-hosting abilities. I just happened to look over and see it. So going back to the slides, do you have the resources, right? So if you're doing a clinical trial, maybe you're the principal investigator and it's just you. You don't have somebody that can do your regulatory or nurse coordination for you. What are the significant risks for the clinical trial? Do you have the patient population, right? So once again, starting with that end in mind, you know, for the patient population, you know, maybe you looked at my chart, did a cohort through there. You might have used Trinetics, things like that to try to find out, you know, do you have the patient population? So let's talk about how is a protocol helpful? So a lot of stuff here. So let's kind of walk through it. You want to have clear outcomes, right? Because you want to make sure that you have that data integrity there, and you want to make sure that when you get to the end of the clinical trial, you're either, you know, proving your hypothesis that you thought, or maybe not, and maybe you need to take the clinical trial in a different route. You want to have a detailed plan, and that detailed plan is someone should be able to pick it up and run with that study. So making sure that everything is clearly thought out, and you're thinking about all aspects of the clinical trial. And it's really, the protocol is really that essential component of the research. Also thinking about the treatment and the safety plan for the patient. You want to reduce that non-compliance, right? So you want to have those details in that protocol, making sure that we reduce that compliance or non-compliance, excuse me. Data collection and analysis scheme. So thinking about ensure the study is conducted, data is collected, it's analyzed, the integrity is scientifically rigorous, and it's ethical and consistent. And also looking at the cost sheet as well. So making sure you're looking at your budget. I did put over here to the side. For some of you, you might be working with a multi-center. So also thinking about if you're doing that, what's that collaboration look like? And that's the thing about a protocol. If you're working with multiple centers, that protocol has to be very clear. So that person at the other center, let's say we're working with Marshall, that they're running that clinical trial the exact same way that we're doing it. Let's talk about protocol's purpose. It's really the roadmap for performance of study. It's really going to give you that next step to operationalize the study. Track changes recommended by the IRB, maybe some clinical monitors or funding agencies. Can also help anticipate problems. So what do I mean by that? I talked a little bit about that the person that was fasting for blood work, but maybe they then also need to you know, do the survey and do the testing. So does that really make sense? Because you have to think about, well, that patient needs to eat. So kind of thinking about just anticipating those types of problems. You know, are you going to have the patient maybe go back to the waiting room to sit and eat? You know, just kind of working all of that out ahead of time. Because you want to safeguard the participants' health and safety. And you also want to facilitate communication among, you know, your collaborators, your employers, your funding agency. So for example, if you have a patient and maybe they have to go over to the cardiopulmonology exercise test, but that is in a different building, how will the patient get there? Will they just arrive at that building? Or do they need to see the study coordinator? So does the study coordinator need to go to them? So just kind of thinking about even the logistics for the patient, making sure that they understand the flow of the study for them as well. And this also, the protocol can also help assist in your manuscript authorization and preparation as well. So let's talk about protocols and standard operating procedures. Studies vary, right? Single detailed protocol, but sometimes you need a protocol and you also need the standard operating procedures behind it. So, excuse me, standard operating procedures are finely detailed information describing the conduct and operation of the clinical trial. So for example, your protocol will state which tubes will be used to collect blood, and it will be shipped to this location for processing. Well, if that's all your protocol says, how do you know how many tubes are to be taken, how it's to be processed, how long does it have to be spun in the centrifuge, what is the location, how are we going to ship it to that location? Does it need to be overnight? Does it need to be ice packed? Things like that. So your protocol might not have all that detail in it. So you might have, going along with that, standard operating procedures as well. Going back to that multiple site study as well, different institutions may have varying protocol requirements. So if you are working with a multi-center site, kind of think about that as well. And using study SOPs is very useful because once again, all that detail is written out there. And you also may have to follow the SOPs of a different institution, or if you are working with sponsors, sometimes the sponsors want you to use their SOPs as well. The other thing is complicated or long procedures. That SOP may be more useful for writing, once again, those details in there. I just want to point out here, and I put all explanation points, I even put 2 explanation points that tells you how important it is. That documents have to all agree. So if you are going to have a protocol and you're going to have a standard operating procedure, if you update the protocol, you have to look at the standard operating procedure to find out if that also needs updated as well. So making sure all of those documents agree. I did put here some resources for for manuscripts or for other submissions. So NIH is here. They have a protocol tool and everybody can register for that. I even registered, and I'm not a principal investigator whatsoever, but you can go in, it kind of walks you through. I do have the templates there for the NIH, for the IND, IDE phase 2 and 3. So that is also, and then I have a couple other ones there as well. And just want to point out too, don't forget the IRB also has their template website as well. So the NIH has electronic fillable protocol, and it also has a Word document that has instructions. So just to let you know that. When I send the slides out today, okay, I'm gonna be honest, I'm not gonna send the slides out today. I'll get them to you by end of week. These will be hyperlinked so that you're more than welcome to go see those resources. So choosing a protocol template. Templates follow similar outlines for topics to address. There's instructional text which explains what to include in each section. Make sure that you're removing that when you're, once you write your text in there, don't leave the instructions in there. Some protocol templates may even include example language or graphics that they're looking for that you might include in that section. And use a template that best fits your study. And you want to customize that template because once again, it is a template. So make sure that you customize that to your research. And please, please, please delete instructions. I've literally seen that where they've left the instructions in, so please delete the instructions. All right, so let's start to write the actual protocol. So my people are all there, they're on the starting line, they're ready to go. You'll have several drafts of the protocol before it's finalized. Give me 1 second, I see a question coming through. Cindy, I saw your question. Lisa's going to be working on that. You have several drafts of the protocol before it's finalized. So don't think just because you write it, you're like, I'm done. It's going to be finalized. That's not always the case. Because you want to write it, you want to review it, and you want to improve it. You can get input from others. That's really suggested to get that input from others. Consult with study team, collaborators, other doctors. I will say involve a statistician early on, right? Because they might see something and they're like, hey, you didn't think about collecting this. I can't run the data if we don't have this element. Discuss with your study coordinators, you know, because like I talked about the patient that has to go different places, your nurses and your coordinators have done studies before, and they might be able to say, hey, you know, it takes a long time to get over to this clinic. You know, we might want to add some extra time here, things like that. Talk to your finance, your budget staff about cost, making sure that, you know, you have all that stuff there as well. Also have your study team read the protocol. They can definitely provide input as well. I will say the other way that you can get collaboration is we do through our PI Academy. We have the ID Lab that meets the first Wednesday of every month. Our next one is on May 6th from 4 to 5. I've actually sat in on a few of those thus far, and they're very, a lot of discussion. You kind of present either what your protocol you've currently written or an idea that you're thinking of writing. And then we have people on the committee that just ask some questions and just start a conversation. They're like, oh, you know, that's a good idea. I didn't think about that, things like that. So making sure that you do that. And if you want information, I apologize, if you want information about that, feel free to email me. That's totally fine. I can definitely get you in contact with the right person. How much detail to include in protocol? IRB and other reviewers, they need sufficient details to fully understand the research plan. Because remember, they can't read what's in your mind. So making sure, you know, do you ever write it, even not even talk about protocol, but, you know, maybe a case note for a patient. I've written a case note before for a patient and I thought it sounded great until my boss came back and asked me additional questions like, oh yeah, that happened. I forgot to put it in there. So just once again, making sure to fully understand. So how do you do that? You provide supporting evidence for conducting the study, sufficient background information to justify study population, describe all study activities, the what, the where, how, by whom is study conducted. All activities should support a primary, secondary, or exploratory aim of the study. So once again, making sure that you're hitting that support for the primary or secondary aim. Phew, that's a lot of components of a protocol, right? So are we going to be able to review all of those today in the last 40 minutes that we have? I hope you're all shaking your heads no, because we're not. So we are going to focus on a couple of them, but just to kind of give you an overview of some components of a protocol. So first I wanted to talk about the introduction. So you have your study subject, your hypothesis. Once again, it should be really clearly identified and briefly described in the introduction. So with that, I kind of want to start down here with the physician. So we have the physician, he or she comes up with, you know, what their clinical trial is. They're going to write the details on what is the, you know, how we're going to conduct the clinical trials in their protocol. And then it goes back, and then the next step you need to kind of think about is analyzing that data, right? Once again, making sure you have that statistician to help you out. If you don't have a statistician on your team, the WVCTSI, we have statisticians. Please feel free to reach out to us. So analyzing that data. And I do have arrows here on purpose because this might take a couple rounds to kind of go through. Because maybe when you get to analyzing data, you realize that maybe you collected data that you don't need. So instead of having the patient go through maybe a test that they don't need to do, go back and rewrite your protocol. So just once again, just making sure that you're really thinking about it. Because you want the rationale, right? You want to understand what is the underlying intervention that you're looking to obtain. The definition of the problem should be clear so a reader can recognize the real meaning of it. So let's talk about the background. Relevant, basic, just preclinical and clinical research. Important literal and relevant data that provides background for the study. So what you're doing here is providing that data supporting your rationale. You might have found, you might identify gaps in literature. So maybe you're looking at chronic back pain and you notice that nobody looked at individuals that were 60 plus years. So maybe that's, make sure you provide that in your introduction. And then of course, any relevant treatment issues or controversies as well. So let's talk about study objectives. So write specific objectives in early stages of development, right? So you're gonna write down that objective. So I have two here. The first one is the objective of the study is to determine the safety and efficacy of drug, let's say Acme, in diabetes, okay? My second one there is to assess the safety, identify possible side effects, and to optimize stimulation parameters of continuous VMH-DSB. Additionally, the study aims to determine if continuous VMH-DSB will lead to weight loss by causing changes in body composition, basal metabolism, or food intake control. Phew, that was a mouthful for the second one, right? So the first one, that first objective is very clear. It's very specific, while the second one has a lot of components to it. And it's not under a single objective. So once again, when you're kind of thinking about your objectives, just making sure that they're very clear and concise. Another component is your endpoints. So your endpoints, let's talk about it. Let's talk about a definition of an endpoint first. I kind of started there first. And some of you may say, Debbie, I could have given you that definition during training. So I'm all about definitions. I like to make sure we're all on the same page with our definitions. And it's an event or outcome that can be measured objectively to determine whether the intervention being studied is beneficial. So thinking about that as we're walking through this. So clinically meaningful endpoints that directly measure how a patient feels, their functions, or survives. The other thing is endpoints that in themselves represent or characterize the clinical outcome of interest. So my objective, is it survival? Is it disease extirpation, clinical event, or is it subjective? I'm looking at a symptom score. Health-related quality of life, making sure it's a validated instrument. So customarily, the basis for approval of new drugs. So making sure that you're including those things in your endpoints. Let's talk about eligibility criteria. So a failure to write eligibility criteria properly can lead to failure to mimic clinical practice, increased study complexity, increased cost, less numbers of patients being recruited, and safety issue. It could lead to a safety issue if it's not well defined and potentially put patients at risk. So making sure when you're looking at your eligibility criteria that you really want to ensure that you're looking at that, making sure that patient is staying safe. So let's look at that a little bit more, and we're going to look at it through study population. So I have my inclusion, exclusion criteria there. So for my inclusion and exclusion, it's really looking at a detail in the balance, right? So that's why I have it on scales there. And no, none of us are getting on scales after we've been quarantined for this long. I don't know about you guys, but I am not getting on that scale on Mondays. I'll deal with that when I get back to work. So inclusion, exclusion details is that balancing act. So you need to think about the hypothesis that you want to test and what is the patient population that you should be testing. So for example, there for my inclusion, I'm going to start at the bottom here. We'll start with my demographics. What's the age range? Is it male or female that I'm looking? You might be looking at, no study has been looked at for chronic back pain for the people over 60 years old. Does that need to be looked at to determine what works for that age of a participant? Looking at disease for your inclusion. So I just said chronic back pain, but what you want to put parameters around that. So you don't want to just say, oh, it's just chronic back pain. You might want to put how long have they had chronic back pain? 2 months, 1 year. You know, what is chronic back pain? Making sure that you spell that out as well. For your inclusion, you also want to look at laboratory results. So you might want to determine if you're doing a diabetic study, you might want to look at their A1C level at baseline. So let's pop to the other side of the scale. This is the lighter side, so I like this side better now. I'm just messing. For your exclusion, once again, looking at your laboratory results, you know, providing that range. If you're looking for that exclusion, make sure you're putting that range there. And then I have health conditions. So I think about the patient in the real world and the safety aspects, because it wouldn't be ethical. So for example, if you have an extreme mental health, standard of care might be better for them instead of being delayed on getting that treatment. You know, maybe you're looking at pregnancy, but you don't know the impact on the fetus. So making sure that that might be an exclusion criteria for them. So think about, you know, the other exclusion is, for example, I was in a clinical trial and I had to attend a session 6 weeks, and it was every Tuesday night. Didn't change, it was every Tuesday night. You know, they had a part of it, they actually videotaped it. So we had the next week, but you know, what does that look like? So just kind of thinking about those kind of things as you're doing your inclusion and exclusion. Study population recruitment. So strategies for recruitment and retention. How will you identify and recruit? You know, think about advertising where, right? Let's go back to our chronic back pain people or individuals, sorry. We're looking at maybe the 60+ range, right? Is social media maybe the best place to get all your patients? Probably not. And it might be a combination of social media, but you might want to do, you know, an advertisement. Obviously, it has to be IRB approved. You might go to WVU MyChart, do a cohort through there, and do a message out through MyChart. I jumped ahead. Where will you recruit? Sorry. How will you document ineligibility and non-participation of eligible candidates? So kind of think about that as well. You know, there's a consort diagram, and that's a simple flow diagram showing the enrollment of patients, their allocation to treatment, disposition status in the clinical trial, and it's a layout of a consort diagram, depends on the study design. That is required for many types of publications, so you would kind of get a little bit ahead on your publication if you already had that in as part of your recruitment. Just want to mention that Encore, not all that information can be placed in Encore, but if the patient was consented and doesn't pass the screening process, that is entered, and then the study coordinator can keep track of what's not entered in Encore, so just putting that out there. Defining your consent procedure. Also define randomization procedure if it's applicable, obviously, for your clinical trial. Study assessments and procedures. So you have efficacy assessments, right? Study procedures and evaluations to support that determination of efficacy of primary and secondary endpoints. So what's the biological specimen collection? What's the assessment of intervention adherence? Once again, going back to safety and other assessments, what's the study procedures and evaluations to monitor safety? So think about physical exams, vital signs, EKGs, laboratory evaluations, questionnaires, because once again, if you have a questionnaire, that person may, you know, have put in, you know, an adverse event. So making sure that that's written in there as well. And of course, your adverse event monitoring. So let's talk about your study design. What does your study design look like? Once again, a lot of information. And once again, I'm not going to be covering that here in the last 30 minutes of our presentation. But just a lot of things to think about, you know, like target enrollment, your number of participants, what's your intervention, your treatment, what's your follow-up look like? What if they have unscheduled visits? You know, we're kind of dealing with that right now, some of us, for COVID. You know, people can't come in, things like that. So making sure that that is part of your study design as well. And I apologize, and if anybody else realized, you can totally tell me in the chat, I realize I have unscheduled visits there twice, so apologize about that. And why do you catch that now? Not, you know, the 10 times you looked at the slides before, it's okay. Statistical analyses. So you want to clearly state all the variables measured in the study with their corresponding baseline and follow-up assessments. So what is the direct measures? What's the source? What's the derived measures? How is it computed? So you want to make sure you have your unit of measure. So for example, if you're doing blood pressure, you want to make sure you have your mmHg there, right? And this is kind of going back to Alcoa, right? Making sure that you have the units next to the measure. You know, systolic blood pressure or diastolic blood pressure, or mean arterial pressure, known as MAP. So if it's MAP, is that direct from the instrument or is it computed? So making sure that you have that in your statistical analysis. How is that going to be captured? Clearly state all the variables relate to a specific arm. So it's your primary, your secondary, exploratory. Will it be used in the analysis plan? So is it efficacy outcome, safety outcome, or covariant? If no clear purpose, why allocate resources and why burden the participants? So kind of going back to the statement I made earlier, if you're collecting information and you may not need it to prove the hypothesis, that statistician may be able to help you work through that. Or maybe, you know, I know the question's that, but the other question could be that you didn't add something that you didn't realize you would need, so. You want a well-developed statistical analysis plans, and it should include your statistical estimates. You know, what's the medians, the proportions, the incident rates, the mean differentials, or excuse me, differences, excuse me, that will be tabulated along with corresponding codefiance intervals, or CIs. Complete list of the null hypothesis, including the outcome measures involved and the details of the test procedure. So once again, if I haven't said it, I think I've said it like 4 times, I believe, if anyone's been counting me. Make sure you consult with a statistician early on in the development of your protocol, because that is a lot, just all those math terms in there, whew, right? So making sure you consult with them. When applicable, you want to complete specifications of the statistical models to be fitted, including your covariance and your assumptions. A reasoned strategy for dealing with the multiplicity of hypothesis tests. Sensitivity analysis to examine robustness of the main results. Distributional assumptions, you know, for your priori considerations. All right, so let's talk about the data safety monitoring plan. So once again, Debbie's going back to her definitions. So what is a data safety monitoring plan? We do refer to it as DSMP. And this is from, I actually took this information from the National Institute of Health. So it ensures the safety of research participants and protecting the validity and integrity of study data in clinical trials. So this document really is the process that ensures the safety of participants while ensuring integrity of data being collected. And every clinical trial really requires some level of monitoring for safety. It may not be a complete DSMP, but there should be some type of monitoring in that clinical trial. And we'll kind of talk about that as we go through these slides. You want to look at the risk associated with participants in research, because you want to minimize that to the greatest extent possible. Hence, we have the methods and intensity of monitoring should commensurate with the risk, nature, size, and complexity of the trial. So you really want to, in that DSMP, you're going to formalize the monitoring process and assign responsibility for monitoring study-specific events on a predefined schedule. So making sure that that's set up in your DSMP. So I have a disclaimer here, and obviously I took this screenshot before COVID because obviously they are not doing social distancing whatsoever. They're all trying to jam themselves into one phone booth. So my point here is we're going to be the superhero because I can't cover everything in this presentation. And I'm going to be honest, this presentation was a lot more slides and we actually had to cut it down just to make sure we had time. So making sure that this is my disclaimer, I can't cover everything. And not everything may apply, but really think about how you can apply concepts indirectly to your DSMP. So let's do study monitoring first. So monitoring of the study. Once again, ensuring that safety of participants. You want to protect the rights of human subjects. Monitoring for safety, occurrence of adverse events, assess continued reasonable risk and benefit ratio, ensures integrity and quality of data. So we have a monitoring trial data for accuracy, completeness, ensure variability, and verify trial conducted in compliance with the protocol, right? Because when you're doing that monitoring, once again, you're going back to the protocol, you're looking at good clinical practices, you're looking at regulatory requirements. So making sure all of that is being done in that clinical trial and that they're following the protocol that they wrote. The current version of the protocol. So let's determine if a DSMP is required or needed. So when do you need a DSMP? So if you're the physician, and I do have a cartoon there, but male or female, is the clinical trial NIH funded? So if it is NIH funded, you do need to have a DSMP. That was decided in June of 1998, the NIH policy for data and safety monitoring. So just to let you know that. If it's not, then we're going to look at the next couple of slides. So first of all, think about your protocol complexity. So I have a DSMP there in the very middle. And one side I'm doing very simple protocol. The next one, the other side to the right, I'm doing very complicated. So if it's very simple, if you're doing survey, if you're doing focus groups, maybe it's a minimal risk observational study, data specimen repository, you may not need a DSMP for that because it's on that very simple part. But once we go more complicated, it's more than a minimum risk. You're looking at a drug or device study. You might be looking at a study with an IND or an IDE. If you're looking at a multi-site study, so once again, going to very complicated protocol, you would want that DSMP. So we determined maybe that we need a DSMP. Now we need to decide how complicated, what's the complexity of our DSMP? Is it very simple, where I'm going to start on the left-hand side, where the investigator is monitoring it? Or is the investigator monitoring and we also have a safety monitor as well? Or do we need a data safety monitoring board? Do we need that data safety monitoring board? I should have stuck with the acronym. And a safety monitor. Or do we need a DSMB comma safety monitoring plus? So it's a very complicated. So kind of thinking about that as you're writing your DSMP. So when do you need a DSMP? So once again, we're going back to that scale of looking at different aspects of your protocol. What is the risk for the patient? What is the population you're looking at? What is the size, the scope and complexity? So you just want to make sure that you have adequate provision to monitor the data collected to once again ensure the safety of the subjects. You also want those provisions to protect the privacy of subjects and the confidentiality of the data maintained as well. So kind of think about that. So let's look at monitoring levels. So PI responsibility, so my principal investigator's responsibility. The PI is responsible for personally conducting or supervising the conduct of human subjects research and for protecting the rights, safety, and welfare of the subjects enrolled in the research. So the PI is ultimately the responsible for all aspects of the trial, including safety. The inclusion of other reviewers does not relieve the investigator of his or her responsibility. So they can't say, well, we have a DSMB, you know, they'll take care of that. Ultimately, that PI still has that responsibility. The PI must ensure that all human subject research is conducted in an ethical manner and in accordance with looking at any federal, state, or local law regulations, their institution policies as well, any requirements of the IRB office. Also thinking about, you know, does it make more sense to have an independent monitor because that independent monitor might have an unbiased view of the clinical trial versus, you know, the PI might have a conflict of interest, you know, especially if that investigator is assuming the role of the monitor. So kind of thinking about that as well. So next thing we need to look at is determining the risk. So we're going to look at low, moderate, and high, or minimum, obviously that's the slide that we're on, low, moderate, and high risk. So minimum risk is the probability and magnitude of harm or discomfort anticipated in the research are not greater in or of themselves than those ordinarily encountered in daily life or during the performance of routine physical or psychological examinations or tests. So for example, if the patient it's just a routine physical test, it might just be peripheral blood draws, routine x-rays, a routine psychological examination, that would be minimum risk, right? So it's not anything invasive for the patient. Next is low risk. So that involves a minor increase over minimum risk. So the intervention or procedure presents experiences that are reasonably commensurate with those inherent in actual or expected medical, dental, psychological, social, or educational situations. So maybe you're looking at studies of healthy volunteers using well-described research procedures, such as you know, muscle and fat biopsy, a low-risk exercise test, an oral glucose tolerance test. So those would be low risk. And next is moderate risk. Are reasonable in relation to anticipated benefits, if any, to subjects and the importance of knowledge that may be reasonably be expected to result. So these risks are recognized as being greater than low, but are not considered as serious as high risk. And their surveillance and protections are adequate to identify adverse events promptly and keep their effects minimum. So we're looking at this, patients may be treated with a placebo for a recognized disease, or a multi-arm study where there is a potential for increased risk in one or more of the arms. You might be looking at, for a moderate risk, you might be looking at an off-label use of an approved medication, or you might be looking at that drug in combination with another drug. You might be looking at low-risk studies, but it's in vulnerable populations. It might be procedures involving the collection of sensitive information, you know, or invasive sampling. So that would be a moderate risk. And last one's high. That involves an intervention or invasive procedure with substantial risk. There is an increased probability for the occurrence of a study-related event that is serious and prolonged or permanent, or there is significant uncertainty about the nature of likelihood of adverse events. So this one, the interventions or invasive procedures that involve that, once again, that substantial risk, might be looking at a blinded phase 1 or phase 2 trial. Studies involving the use of a chemical, a drug, a medical device for which there's little or no human toxicology data. investigator-initiated phase 3 or multi-center clinical trial. So once again, that high risk. And we also want to determine what is the appropriate monitoring levels. So if you have an investigator review, is it a small number of patients? Sorry, let me take a step back. So we have, if we're going to do an investigator review, it might just be a small number of patients. It's only conducted at one site. You have a range of possible study events that could have an important impact on the risk and benefits to research participants. It's very narrow. You might look at an independent review. So looking at that unbiased review. So this is what I talked about, maybe having an independent study monitor, or there was even a suggestion of like 2 or 3 members of independent investigators and biostatisticians. Or you can have what we talked about previous, which is the Data Safety Monitoring Board. And that could be a group of scientists, physicians, statisticians, and others. That large number of subjects where risk may be better assessed through statistical comparisons of treatment groups. If you have blinded study treatment groups, multiple clinical sites, high-risk interventions, and controlled trials with mortality or major morbidity as a primary or secondary endpoint. So continual monitoring and oversight, so I just kind of put it in a graph, or not graph, but a visual here. As some of you know me, I'm very visual. So here I have like a low risk monitoring. Who monitors it might just be the PI, IRB. When to monitor, it might be initial and annual review. Once again, we're doing low risk. You know, you have enrollment, you have withdrawals, you can have AEs and SAEs. Or if it's high risk, you're looking at who's doing the monitoring. Might be a medical monitor, a safety committee, or a DSMB. And it's every so many subjects, it's every 4 months, or an interim analysis as well. What they're looking to monitor is the efficacy, the safety endpoints, the adverse events, and adverse events and serious adverse events, and stopping points. You know, what is the stopping point? So elements of a DSMP. If it's NIH funded example, so I just have that example there. If it's NIH, that's an example of the elements that would need to be in your DSMP. The other things you might want to consider is the plan for protecting confidentiality. So handling of case report forms, such as a secure location with limited access. So if I know that Peggy Sue has a case report form, and I know that she places them in the bottom desk drawer, and she locks it every night when she leaves, and she places her key on her bulletin board, is that limited access? No, because I know it and I'm not on the clinical trial. So that would not be limited access. So once again, we want to make sure we have a plan for protecting confidentiality. Also kind of thinking about housing of the randomization codes, because we have the patient's names with them, right? Where are they going to be stored separated with restricted access again? All study documents will only house coded numbers. So once again, just making sure you have that limited and or restricted access when you're thinking about protecting that confidentiality. So options for developing a DSMP. You can incorporate it within your protocol. You can document that within the IRB application, or you can attach as a separate document to the IRB application. Data management plan. So you have basic elements, data security and confidentiality, data quality, so looking at accuracy, completeness, or missing data. Role responsibilities, develop or maintain the database. Create the code book, enter the data, verify data accuracy, and create and review queries, any questionable values that you're looking at. All right, then you're at your finish. You're at your finished protocol, right? So once again, just takeaways. Just remember that the first protocol you write may not be what you end up with at the end. You know, there's going to be some revision there because you want to make it the best protocol. And even after you finalize it, there could be amendments to it as well. Getting, you know, that statistician in early to help you walk through that and determine if you need that data safety, what level of data safety monitoring you need as well.